Hello, welcome to the online fungi PowerPoint presentation. So today we're going to go over fungi. Fungi are hugely important in ecosystems. They're often overlooked, but their importance cannot be overstated. So they're really important in food webs, and so one of a cool food web story was trying to preserve the keystone species of the spotted owl. So the spotted owl was becoming endangered. So what they started to do is look at the organisms that the spotted owl ate because they need their food supply to survive. So they found out that probably one of the reasons that the populations were diminishing was that this flying squirrel, those populations were also diminishing. So again, they looked at the food source for the squirrel, and they found that the main food source for the squirrel are these underground mushrooms called truffles. So they figured out that the reason that these truffles were disappearing was because old growth forests were being cut down. So this illustrates that this whole network is reliant on each other, and if you are to remove one aspect of this food web, then the whole food web will collapse. They're also seeing a lot of interactions with birds and fungi. One of the really cool ones, there's many cool ones, but one of the cool ones is between woodpeckers and fungi. And so woodpeckers peck wood, as you know, and so they make cavities in trees. And so there's adaptations that woodpeckers have with certain fungi. And so they take specific species of fungi and they carry them to their new habitats on different locations of their body, so either on their beaks or on their shoulders, sometimes on their feet, and they transfer that fungi into the tree that they begin pecking, and then with the help of the tree, they're able to facilitate that cavity building so they can build their homes. We also like to talk about superlatives in biology, so we always like to talk about the oldest and the biggest, and so currently thought of the biggest organism by space is the humongous fungus, which is an armillaria species in Minnesota, and then, Michigan. And they also discovered that this one individual, the humongous fungus, Armillaria gallica here, takes up 37 hectares, and they believe it to be about 2,500 years old. So it most likely might be, well, except for those bristlecone pines, some of the oldest organisms alive. Another important aspect of fungi is their ability to sequester carbon. So as you know, we continue to increase the amount of carbon dioxide that we release into the atmosphere. And a lot of folks are touting planting trees in order for plants to absorb that carbon dioxide, but really the majority of carbon dioxide that is taken out of this atmosphere is held in fungi, particularly in the northern hemisphere. So, moving on to more biology stuff. Here's that phylogenetic tree that we've been looking at for this semester. And remember, all these yellow things are those protistans that we looked at. Over the last few weeks, we've been looking at land plants, and now we're going to move into fungi, which are down here. So, it's not totally inherent to think of fungi as sister to animals. But here are our fungi, and you can see that one time in the not so distant past, they shared a common ancestor with animals. So in your book, it talks about fungi showing up on the scene about 450 million years ago. One of the reasons for this number is we really don't have a lot of fossilized fungi to work with because their body form doesn't lend itself to becoming fossilized very easily. But quite recently, in the last year, they found this fossil, which they believe to be fungi, and that it's 1 billion years old. So a lot of the evidence that we have about the colonization of land, plants were the first organisms to colonize land, and that was only possible in conjunction with fungi. So the more fungal evidence that we're able to gather, the more it points to that fungi evolved long before plants, and it continues to give us evidence to support the hypothesis that the only way that plants were able to make that transition onto land is with some symbiotic association with fungi. So what are fungi? To understand fungi, we want to think about their characteristics. So if we think about their body, some of them are yeasts. So these are single-celled fungi, and you can see these little green globs coming off that SEM are little baby yeast coming off, budding yeast cells. But the vast majority of fungi live their life cycle in a hyphal stage. So hyphae are these long filamentous strands, and that's their body. That's how they eat. So some of the hyphae have cell walls, so we call that septate. Some of them don't have cell walls, which are cenocytic, or aseptate, A-S-E-P-T-A-T-E, A meaning without. And then, just like we looked at plants a couple weeks ago, fungi have specialized structures. So some of them have these cool structures where this is a nematode, so it's a little roundworm that cruises through the soil. Some fungal hyphae have these structures that can actually grab onto these nematodes, trap them, and then slowly digest them. And then we'll talk about mycorrhizae. So there's some fungal structures that differentiate off the hyphae so that they can associate with plants, and they have different relationships with plants as far as how they transfer nutrients and between each other. A group of hyphae is called mycelium. So if we look at this white mass here, all these thin little strands are individual hyphae. Collectively, we call this mass mycelium. Fungi have cell walls of chitin. So if you ever stepped on an insect and heard that crunch, that's chitin. So that's one of the lines of evidence that is supportive of the fact that fungi share a common ancestor with animals. If we think about other storage molecules, we have starch and cellulose, which are storage molecules of plants. And then there's also glycogen. So you can see that these are all basically glucose monomers attached in different configurations. And then fungi have a nutritional mode that we haven't really discussed before. They eat via this mechanism of absorptive nutrition. So they're heterotrophic. They cannot make their own food. And what they do is all these little purple dots that are coming out of this hyphal strand are enzymes. So the enzymes are excreted into the substrate. They digest their nutrition extracellularly outside of their body, and then they kind of slurp it up like a little smoothie. They are either saprophytic, which means they break down dead organic material, or they are symbiotic, meaning they live within a living organism. So this is the overall fungal reproduction life cycle. Some fungi reproduce only asexually, some fungi reproduce sexually, and some reproduce with both mechanisms. So we're going to start down here where we have the release of spores. So the spores in the right conditions will germinate. For those fungi that can do either pathway, there are probably some signals that will send them on either pathway. They're going to germinate. For the vast majority, they're going to germinate into hyphae. A group of hyphae, again, is called mycelium. And they're going to differentiate into some spore-producing structures, meaning they just make some hyphae, then they wall off those hyphae into spores, and those spores disperse into other habitats. Now, if they're going to undergo the sexual reproductive cycle, they have to find another hyphal strand that they can mate with, and they'll fuse. And once they fuse, there's a hyphal strand with different types of nuclei inside it. That fusion of the hyphae is called plasmogamy. So gamy means fusion and of the plasma. So cytoplasm fusion. So at this point, they have a heterokaryotic stage. So hetero means different. Karyo, if you remember from eukaryotic and prokaryotic, karyo is translated, its literal translation is nut, but it means nucleus. So in this stage, they have different nuclei, hetero different karyonuclei. Then at some point, those nuclei, karyo, are gonna fuse, gamy. At this point, we have a structure that is diploid. So those two haploid nuclei have fused, they're gonna undergo meiosis, and then we're gonna have haploid spores. So we'll go through this fungal reproduction in more detail in the specific lineages that we talk about. So today we're going to talk about 5 lineages: the Chytridiomycota, the Zygomycota, the Glomeromycota, the Ascomycota, and the Basidiomycota. For Zygo, Asco, and Basidiomycota, the easy way to remember those is that that's the reproductive structure. The chytrids are completely different from the rest, and then the Glomeromycota have no sexual reproductive stage that we know of. So those will be how you can distinguish those guys. So the first group that we're going to talk about are the chytrids. You'll notice that this is a polyphyletic group. We talk about them as chytrids because they have this one structure that's similar between all of them. They have a flagellated life cycle stage. This is unique to the whole kingdom of fungi. No other group of fungi have flagellated life cycle stages. So automatically you should be thinking about how we saw this type of structure in plants. What medium do these organisms need to be able to reproduce? Since they're flagellated, we know that they swim, so they need some sort of aquatic system so that this flagellated life cycle stage can move through the environment. Just like we saw in plants, the more derived lineages are going to lose this flagella. That is an adaptation for terrestrial habitats. The big thing that you should know about chytrids is a disease that they cause called chytridiomycosis. So in this, the chytrid fungus infects the ectoderm, the skin of an amphibian. So what you can see is in this life cycle stage, these zoospores, these are the flagellated life cycle stage, they're going to produce a structure. This structure embeds in the skin tissue of an amphibian. Now, if we think about this, what types of habitats do most amphibians live in? They're semi-aquatic, and the skin provides a function that our skin does not provide. Skin, right, is for protection, but amphibians also use it to breathe. So that's why most of them spend a lot of their time near water, because they need some sort of moisture in order to do gas exchange across their skin. So if we start to infect their skin, they're going to break down their ability to do gas exchange, ultimately suffocating. So these fungi are rapidly differentiating and attacking many, many different amphibians. So this is going to drastically reduce the number of amphibians and amphibian species in the next 50 years. It is said that a recent paper showed 151 amphibian species were impacted by different species of chytrids, and there are presumed to be 90 species extinctions due to this chytrid fungus. Different species of chytrid fungi. All right, the next lineage that we'll look at are zygomycetes, and again, you can see this is polyphyletic. Zygomycetes reproduce sexually via zygospores. So this is the first organism that will look at or the first fungi that will really look at their life cycle stage. So if we come down here, we're going to start with a sporangium, which produces haploid spores. So in some cases, the spores can germinate into mycelium. This is a single strand, so that's a fungal strand, and all the little red dots are nuclei. What you'll notice is that there aren't any cell walls in the zygomycete hyphae. So, given some sort of reproductive stimulus, an asexual reproductive stage called a sporangia will form, and the nuclei will basically be walled off into spores. These spores can then be dispersed and germinate. So if we look at over here, they can either go into this asexual reproduction route ad infinitum, or they can enter the sexual reproductive route. So here we have spores, and here we have two different hyphae germinated. You can see that this one has red nuclei and this one has blue nuclei. So if they decide that they're a mating compatible type, they're going to produce these structures that are going to fuse. At the end of these structures, they produce these structures called gametangia, and these gametangia are going to fuse. So if we zoom in microscopically, you can see one gametangia and one gametangia here. This structure is going to fuse. So that's plasmogamy, right? The cells fuse, the cytoplasm fuses. Plasma, cytoplasm, gamete fusion. And here we have that structure. So inside this structure, it's one cell, and it has multiple different types of nuclei. So it's hetero, different, karyotic, karyonuclei. At some point, these nuclei are going to fuse. That is the process of karyogamy. So karyonuclei, gamete fusion. At this point, this is a relatively short-lived stage. We have diploid nuclei. Those diploid nuclei are going to undergo meiosis, and this zygospore, this is the zygosporangia, now we have a zygospore. This zygospore is going to germinate, produce a sporangia, and those spores are going to disperse. So zygomycota reproduce via zygospores. And you've seen this all. That black furry stuff that you see on your bread, that is a zygomycota. Things that are called black molds generally are in this group, zygomycota. So if we were to look under the microscope, the asexual reproductive stage is the sporangia, which produces spores, and then this is the sexual reproductive stage. So inside here are where it's heterokaryotic. Those nuclei are going to fuse. We're going to have a diploid nuclei stage, and then very rapidly they're going to undergo meiosis, then eventually this zygospore can germinate. You also notice on this slide we've got, a little sporangia in there too. So don't be confused by that. All these darkish structures, those are all our zygospores. So here they are developing. Now, think about how these zygospores are dispersed. We've moved away from water, so these are all wind dispersed. So if you think about this, think about where these things might end up on you. They're very, very small and they're wind dispersed, so these are the things that you can breathe in. So folks who have mold allergies, this is often the thing that they're allergic to. There's different chemical compounds in these walls, and you can just see that from a mechanical standpoint, this could be really irritating. All those different spines and things. Also in this group is this funky little fungus called Pilobolus. So this structure here functions like an eyeball. It actually can refract light, and there's a little light sensor at the base here. So this whole structure is going to orient itself so that it's facing right at the light. On this tip here is the spore mass. So what happens is it focuses towards the light. This spore mass flies off, sticks to a blade of grass, and then that blade of grass is consumed by an herbivore. We all know what happens when we process food. Something comes out the other end. So this spore mass comes out the other end of the herbivore, and it's ingenious, right? It's got this fertilizer packet. But most herbivores are not going to eat their own feces, so it's got to figure out a way to get the spore away from the feces. So this is what this looks like in action. So what's happening here is you can see this ring right here, that's all water. This fungus has built up so much water to make this hydrostatic pressure that this spore mass is shooting off. It is shooting off so fast that this fungus is the fastest accelerating organism on the planet. Some of my colleagues have looked into this further, and what's really interesting, if you look at this photo right here, that's a little roundworm. So if you've ever had pets and you've had to deworm them, that's what you're deworming them from, roundworms. So what this little roundworm is doing is climbing up onto this pilobolus, hanging out on that spore, and waiting to get shot off. So this nematode is a pathogen for herbivores, and it has figured out how to disperse itself, because these guys also come out in the feces of herbivores.